The cornea on the front surface of the eye is like our window to the world, and it's important that it remains transparent. The outermost layer of the cornea is called the epithelium, and this is like the outer layer of skin, so it's constantly being sloughed off when we blink and needs to be regenerated throughout life. And this happens as a result of the activity of a population of stem cells which live around the outside edge of the cornea. If these stem cells are compromised by disease or injury, such as a chemical burn, then patients suffer from a condition called limbal stem cell failure, and this results in the cornea becoming scarred and opaque, so they lose their vision. They get lots of blood vessels growing across the front of the eye and inflammation. This is also a very painful disease. Clinically, how we're trying to help these patients is by growing their own stem cells in the laboratory and transplanting them onto the front surface of the eye. This involves taking surgically a small biopsy from the patient's healthy eye, assuming that only one eye is affected by injury or disease. That tissue comes to us into our clean room. We isolate the cells through a series of enzyme digestions. We grow those cells up on a substrate which will then allow us to give those back to the surgeon. This process takes about 3 to 4 weeks. After the patient has had their surgery, after a couple of weeks, they can hopefully start to expect to see some improvement in their vision. These round purple areas here are actually colonies of cells, so it's lots of cells grouped together which have arisen as a result of the division of just one stem cell. And we know that we're looking at stem cells if the diameter of these colonies is greater than 2 millimeters. So this culture dish would have been seeded with 1,000 cells, and each one of these large colonies that you see here has been established from just one stem cell. So far we've treated 20 patients. Some of those have had their own cells, others have had cells from donors. Of the first 10 patients that we've treated, 60% of those have experienced improved vision and a reduction in their pain. The second cohort of 10 patients we are following up. Our longest follow-up time, successful follow-up time, is 3 years.
This is like a dead culture of the stem cells. And what I'm trying to do is to translate these into like a 3D structure. So I take pictures of these stem cells and tissue. So I put this in the computer and program, and the computer do like a mesh out of these, and it's like a translation of these cells. These are the 3D structures generated by the computer, and this is like a microscope picture of these stem cells. So I'm trying to take the matrix of these and translate it into these. Ideally, these 3D structures that I'm doing from the structure of the stem cells would be used for design materials, more resistant and efficient materials. There is like a second part of the project that is concerning the way nowadays we see technology. In the mid of the 20th century, we would start to have this ideal robots that would be much better than us, and we would replace, decay parts of bodies by these metal pieces, like highly mechanized and with high technology. Nowadays, these stem cells research, it's just replacing flesh by flesh. So you replace like the pathologic cells, the bad ones, with your own healthy cells. And all this could be almost like your life insurance later on. So that's going to be the second part of the project. So what was considered like disposable, it can be like reusable and precious now.